Hi everyone, welcome to my biology review for the ATITs. This is part 3. If you haven't watched part 1 and 2, that's totally fine, but there are a few topics mentioned in here that will be easier to understand if you have the knowledge from the previous videos. So with that being said, today we're going to go over what a cell is, what kind of types of cells there are, and the structures of a cell and the most important organelles that you're going to need to know for your ATITs exam. So let's go ahead and dive in and get started. What are cells? Cells are the basic building block of all living things, and cells also have the important job of carrying around DNA for all living organisms. So because cells make up basically all living things, the cell theory is so significant because it states the inevitable roles of the cells themselves. So it comes in 3 parts. The first is that all organisms are made of cells. The second is that cells are the basic unit of all living things. And the third is that cells are made from pre-existing cells. Keep in mind that this definition of modern cell theory is super simplified. It's basically a general understanding of what it is. So if you look through a textbook or another resource, you might see a very extended and detailed version of the modern cell theory. However, these 3 sentences basically just break it down. So now what kind of cells are there? Well, all cells of living things fall into one of these 2 categories. Either the cells are prokaryotic cells or eukaryotic cells. So when we say prokaryotic, we're referring to the cell itself or the type of cell itself. When we say prokaryote, this is referring to the organism that is made up of prokaryotic cells. When we say eukaryotic, this refers to the cell itself. And when we say eukaryote, this is referring to the organism that is made out of eukaryotic cells. So your prokaryotic and eukaryotic cells actually have a lot more in common than they do differences. Nevertheless, they do have one major difference. So prokaryotic cells lack a nucleus and a few other organelles. However, they lack a nucleus, and this is the big takeaway. Your prokaryotic cells include bacteria, archaea, and cyanobacteria. So in contrast to eukaryotes or eukaryotic cells, they have a nucleus. This is the major difference between the eukaryotes and the prokaryotes. So eukaryotes have a nucleus, and these include your plants, your animals, and your fungi. So we're going to focus in on our eukaryotes because mainly when we talk about cells, we're talking about eukaryotic cells. Like when we're talking about the human cells, they're all eukaryotic cells. Correction. So all human cells are eukaryotic cells. However, there are colonies of bacteria that is normal in our stomach and our digestive tract, and these are known as like probiotics. So these bacterial cells are prokaryotic because remember, prokaryotic includes bacteria. So let's talk about the differences between plant cells and animal cells. So plant cells have a cell wall that surrounds their cell membrane. Both plant and animal cells have a cell membrane that surrounds their cell and protects it. However, plants have an extra durable cell wall that is made out of cellulose, and this just adds extra strength and extra protection to the plant cell. So another major difference between plants and animals is the way that they get their food or energy. Plants are what you call autotrophs, and animals are what you call heterotrophs. So autotrophs make their own food. Plants and algae are great examples of autotrophs. They basically take the energy of the sun and they transform it into glucose, which is the food source and energy source for these plants. And this process is called photosynthesis. So humans and animals, on the other hand, we cannot do photosynthesis. So we are called consumers, or heterotrophs are called consumers because they must consume something else in order to get their food and their energy. So these are the major differences between plants and animal cells. And remember that both plants and animal cells are eukaryotic cells. So now let's talk about the similarities between the two. Both plants and animal cells have what we call a membrane-bound organelles. So what this basically means is that each organelle within the cell has its own protective layer surrounding it. So they have something called a phospholipid bilayer, which we'll go ahead and take a look at a little bit later. But just keep in mind that this protective phospholipid bilayer is used to regulate what comes in and out of each organelle. There is one exception to these organelles. The ribosomes are the one that do not have a phospholipid bilayer, so ribosomes are not membrane-bound organelles. Okay, so secondly, plants and animals, they both have the main organelles like your mitochondria, nucleus, endoplasmic reticulum, the Golgi apparatus, and peroxisomes, and they both also have the cytoskeleton. So notice that I left out the lysosomes, and that's because yes, both animals and plants do have lysosomes. However, it is far less likely to find them in the plant cell. So now we know what a cell is, what type of cells there are, the differences between eukaryotic cells and prokaryotic cells, and the differences between plant and animal cells. So now let's go ahead and look at the structures of a cell, in other words, the organelles in a cell, starting off with the cytoplasm. Your cytoplasm is everything that is within the cell walls. So all of the organelles within the cell walls and also the fluid that surrounds these organelles, this is all part of the cytoplasm. There's also something called the cytosol. Cytosol is all of that matrix jelly-like liquid substance that surrounds all of the organelles. So remember, cytoplasm and cytosol. Your cytosol is within the cytoplasm or is a part of the cytoplasm. So next we have our cell membrane or our cell wall, and it's composed of this phospholipid bilayer. Bi meaning two, so it's a two layers or double layers of phospholipids. In our phospholipid bilayer, we have a polar side and a non-polar side. The head are polar meaning that they are able to interact with water. They are water loving. So water loving is called hydrophilic. So this head interacts with water, and it's important that it's on the outer layer of this phospholipid bilayer because we have water on the inside of the cell and water on the outside of the cell. So it stays on the outer layer of this phospholipid bilayer so that it can easily interact with the molecules within the liquid or within the water. So the head is polar and hydrophilic, while the tails that are hiding on the inner side, these are non-polar. They do not like the water and they do not react well in the water. So these are called hydrophobic. So the cell wall is a semi-permeable membrane that acts as a barrier. It carefully selects what can come inside the cell and what can leave the cell. There are some really important molecules that hang out around this phospholipid bilayer, and one of them is cholesterol. Cholesterol regulates the fluidity between the phospholipids, and fluidity is basically referring to the space between these phospholipids. The more space there is between them, the better chance molecules have of getting into and out of the cell. Now, this can be harmful if the space or the gaps are way too large between these phospholipids because it will be harder for the cell membrane to control what comes in and out of the cell. Now, in contrast, if there is very little space between these phospholipids, then it will be harder for bigger molecules to get in and out of the cell membrane. So having too much space or too little space is both harmful to the cell. Temperature has a lot to do with this. So the lower the temperature is, the more compact and rigid these phosphomolecules are going to be. Now, how this works is the cholesterol is going to come in and act as spacers and space out these phospholipids so that it is easier for molecules to come in and out. Now, the opposite happens when the temperature is too high. When it's too high, then these phospholipids become way too spaced out and they need cholesterol to come in and kind of squeeze them back together. So basically, cholesterol controls fluidity. When it is too cold or the temperatures are too low, the phospholipids are going to try and become too compact and close together, and the cholesterol is going to come in and act as a spacer. When the temperature is too hot or too high, then cholesterol is going to come in and squeeze these phospholipids together so that there's not too many gaps between them. So your second important molecule that hangs out here in the phospholipid bilayer are your proteins. Now, there are two very important proteins, your peripheral proteins and your integral proteins. Peripheral proteins hang out in the peripheral. They don't go into that inner hydrophobic area. They hang out on the outside. And these help in transporting materials in and out of the cell. Now, your integral proteins help with signaling and communicating, as well as moving molecules through the plasma membrane. So just remember here, guys, that we're talking about your cell membrane when we talk about the phospholipid bilayer. And also remember that the most important molecules that are embedded within this phospholipid bilayer are your proteins and your cholesterol. So next we have our nucleus, and it has a few important components to it. First, we have our nuclear envelope that surrounds our nucleus. The nuclear envelope is two layers. The outer layer has ribosomes and the inner layer has lamins. Lamins is a type of protein that has many important roles, but one of the most important is it plays in cell division. Now, the outer layer that has ribosomes, well, we're going to go over ribosomes later, but one thing that's very important is that inside of the nucleus, mRNA is made, and then the mRNA travels to a ribosome, and the ribosome carries it to the rough ER. So a part of your nuclear envelope are these pores, and these pores are made out of proteins. And basically the pores help regulate what comes in the nucleus and what goes out of the nucleus. So next, within the nucleus, we have this thing called a nucleolus, and it's a tiny little sphere that is the site of our RNA synthesis. In other words, our RNA, ribosomal RNA, is made here. And remember that our RNA has such an important role in protein synthesis. Remember, so we'll go over that when we start talking about the ribosomes. But remember, our RNA synthesis is right here in the nucleus. So lastly, we have something called chromatin. Chromatin actually looks like this. It's more like squiggly and loose little strings that are inside of the nucleus, and they're made of DNA, RNA, and proteins, specifically histone proteins. Chromatin is a material that makes up chromosomes. So chromosomes are made out of chromatin. So that's it for the nucleus. So just remember, it has the nuclear envelope, the nuclear pores made out of proteins. You have your lamins inside of the inner layer of the envelope and your ribosomes on the outer layer. You have your nucleolus, which is the site of our RNA synthesis, and then you have your chromatin, and your chromatin is what makes your chromosomes. So let's go ahead and move on to the rough endoplasmic reticulum. So the rough endoplasmic reticulum, or rough ER for short, is covered with ribosomes, and this is its main characteristics. One important thing that happens within the ribosome specifically is translation. Remember, transcription and translation. Transcription is when DNA makes mRNA, and translation is when mRNA translates or converts into amino acid chains, which is a protein. So translation is part of protein synthesis, and this happens specifically in the ribosomes in the rough endoplasmic reticulum. So once these proteins are made, the rough ER helps fold the protein, and it also packages the protein into a vesicle and pushes it out towards the Golgi apparatus. And remember, a vesicle is basically like a packaging box that keeps the molecules safe. The rough ER also participates in a process called glycosylation. Glycosylation is the activation of a protein. So basically it takes these enzymes that are within the rough ER and it adds some sugar residue to the protein, and this activates it. So rough ER, it is the site for protein synthesis, specifically in the ribosomes. It folds the protein, it packages the protein to be pushed out within a vesicle, and it also is a site of glycosylation. Next, we have the smooth endoplasmic reticulum. It's smooth because it has no ribosomes, but it does have a ton of enzymes. And these enzymes help make or create lipids. Lipids like fatty acids, phospholipids, and cholesterol. So we can say that the smooth ER is a site for lipid synthesis. It also makes an enzyme that is super important and found in the liver. It's called CY450. It's found in the liver because it aids in detoxification. Lastly, the smooth endoplasmic reticulum is able to store calcium. So it basically has these little pumps, and calcium is stored here. Whenever calcium is needed in the blood, then these pumps just push out a little bit of calcium at a time for us to use. Next, we have our ribosomes. Remember, ribosomes are found on the rough ER, and that's what makes them rough. So these ribosomes are called the membrane-bound ribosomes. They're also found floating around freely in the cytosol. So these are called free ribosomes or cytosolic ribosomes. So ribosomes actually look a little more like this. They're not just like one flat circle. They have two parts. They have a large top and they have a smaller bottom. Most distinctly, the ribosomes are known to be the site of protein synthesis. And I'm not going to go into too much detail with this because I went into detail in my last video about protein synthesis. So if you're a little bit lost, go back to my last video and check that out. Basically, what happens here is translation. And remember, translation is the second part of protein synthesis. It's when mRNA goes into the ribosomes and it is translated into amino acids. And remember that amino acids are what make your proteins. So translation here happens in the ribosomes, and it is the site of protein synthesis. The Golgi apparatus, first off, it receives vesicles from the smooth ER and the rough ER. And remember, what's coming out of the smooth ER is the lipids, and what's coming out of the rough ER are the proteins. So proteins and lipids are being delivered to the Golgi apparatus. Here, the Golgi apparatus modifies these proteins and lipids using processes like glycosylation and phosphorylation. So once it does this, it packs them up into vesicles, both the protein and the lipids, and it sends them off where they need to go. There are a few parts to the Golgi apparatus that are important to remember, and one is the cis phase. The cis phase is the part of the Golgi where molecules come inwards into the Golgi. So molecules like proteins and lipids are going to enter through the cis phase. Once they're inside of the Golgi, they go through things like glycosylation, phosphorylation, which are what we call modifications, and then they get packaged into vesicles. Once they're packaged in the vesicles, these molecules leave the Golgi apparatus through the trans phase. So just remember, the cis phase is molecules entering the Golgi, and the trans phase is molecules leaving the Golgi. Next, we have our lysosome. Our lysosome is kind of like a cleanup crew. It breaks down macromolecules. Remember, our macromolecules include protein, lipids, nucleic acids, and carbohydrates. Lysosomes have special enzymes that can break down each of these specifically. So, for example, protease is going to break down our protein. Lipase is going to break down our lipids. Nuclease is going to break down our nucleic acids, and glycosidase is going to break down our carbohydrates. The lysosome is also able to break down old, worn out organelles, as well as cells that are no longer able to function. Basically, they're also old and worn out. Autophagy is when the lysosome takes the old, worn out organelles and breaks them down. And autolysis is when the lysosome takes the old, worn out cells and breaks them down. So autophagy and autolysis, this is something that the lysosomes also do. Next, we have the peroxisomes. Peroxisomes help defend us from free radicals. It does this because it has enzymes, and some of the most important enzymes that it has is catalase and oxidase. These metabolic enzymes help convert or break down these free radicals and turn them into not so dangerous molecules. So, in this process of breaking down these free radicals, peroxisomes excrete hydrogen peroxide, so hence the name peroxisome. Next, we have the mitochondria. The mitochondria has two important components to it, the outer membrane and the inner membrane. So our outer membrane has an increase in permeability. Permeability means the ability for things to come in and out of it or through it, right? So an increase in membrane permeability means that it's going to be easier for molecules to come in and out of there. Secondly, we have the inner membrane where there is little or less permeability. So it's going to be harder for molecules to come in and out of the inner membrane. So outer membrane, increase permeability, inner membrane, less permeability. Okay, so there are a couple functions to the mitochondria. The mitochondria, one really cool fact is that it has its own mitochondrial DNA, and this comes from the mother. And secondly, it has a ton of metabolic reactions. But we are not going to go over all of these metabolic reactions because this would take forever, but just know that it has very important metabolic reactions. One other very important thing is that it is the site of ATP synthesis, and that's why we call the mitochondria the powerhouse. It generates most of the chemical energy needed to power the cell's biochemical reaction. So it makes ATP, and this is what gives the whole cell energy to perform or to function. Okay guys, so lastly we have our cytoskeleton. The cytoskeleton is kind of like the glue that keeps everything together inside of the cell. We have three important types of cytoskeleton. One is the microfilaments or the actin, secondly is the intermediate filaments, and third is the microtubules. So first let's talk about microfilaments. Microfilaments or actin help in muscle contraction as well as they control the shape of cells. So this is how they do it. If you recall, during muscle contraction and relaxation, actin works with myosin to contract and relax the muscle. So in the same way that actin helps this contraction, it also helps a single cell kind of contract in a way. So let's just say if a cell needs to be split into two or split apart, actin is going to kind of go around it and constrict it, kind of like a waist trainer, basically. It's going to constrict it so tight and squeeze it so tight that it's going to make the cell split into two. Also, let's just say a cell wants to squeeze into a tight space. Actin is going to come and surround a part of the cell, kind of like a little waist trainer, and it's going to squeeze it and shape it into a way that it can fit through the small space. So basically, actin helps in muscle contraction, but it also forms a ring around cells so that it can shape it to go through small spaces, but also it can help split the cell in half. Next, we have the intermediate filaments. These are stiff and rigid type of fibers, and they are going to help anchor your cell to your cell, as well as your organelles inside of the cell to the cell, as well as the cell to the extracellular matrix. Remember, extracellular matrix is all of that fluid or jelly that surrounds each cell. So basically, intermediate filaments attach all three of these things together so that they're not just floating around everywhere getting lost. Lastly, we have our microtubules. So these microtubules have an important protein called motor proteins within them. These motor proteins are going to help with movement. So they help with intercellular transport, cell division, and cell extensions. This is how. First off, the microtubules are able to bind themselves and attach themselves to molecules. Once they bind themselves, they're able to transport the molecules from one place to another within the cell. Secondly, they help in cell division, specifically the metaphase, because the microtubules attach themselves to the chromosome, sort of like Legos. They are able to attach, but also make a split and separate these chromosomes. And lastly, these microtubules form some cellular extensions. So cellular extensions are things like cilia and flagella, things that need to, beat back and forth really rapidly. They're going to need these motor proteins within the microtubules. Alrighty guys, so that concludes this video. Just remember that all of these listed are the important organelles that you're going to need to know for your TEAS exam: the cell membrane, plasma membrane, cytoplasm and cytosol, nucleus, smooth and rough ER, ribosomes, Golgi, lysosomes, peroxisomes, mitochondria, and the cytoskeleton, which includes your microfilaments, intermediate filaments, and your microtubules.